Hi everyone. Today we become to our main objective for studying plant pathology. We have to know disease control strategies. We know really that plant disease have caused severe losses to humans in several ways, starvation and uprooting of families. For example, this resulted from the Irish famine caused by potato late blight. So the goal of plant disease management is to reduce the economic losses and damage caused by plant disease. These strategies include exclusion, eradication, host resistance, protection, therapy, and biological control. What is very important to know, plant disease management practices depend on a way that prevent disease from occurring. Therefore, correct diagnosis of a disease is necessary to identify the pathogen, which is the real target of any disease management program. Also, you must take disease triangle into consideration because if you break any side of triangle, that means stop of disease development. We have 5 or 6 strategies used in disease management include exclusion that apply to prevent or avoid the introduction of inoculum in new area. Eradication and means eliminate, destroy or inactivate the inoculum in infected plant or infected area. Host resistance, these cultivars that are resistant to or tolerant to infection. Some modification of culture practices alter environment to be less favorable to disease. And we can use some protection or therapy treatment that prevent infection or cure plant that are already infected. Now we have to talk about each strategy and how can apply in the plant disease management programs. Exclusion is defined as any measure that prevents the introduction of a pathogen into uncontaminated region. That means that disease management measures applied before infection. An example of the first principle is enforcement of quarantine to prevent introduction of a pathogen into a region where it doesn't occur. Exclusion measures usually only delay the entry of a pathogen, also may provide time to plan how to manage the pathogen when it arrives. The second principle for excluding pathogens is to produce pathogen-free seed or planting stock through certification programs for seeds and vegetatively propagated plant materials such as potato. Planting stock that is free of pathogen can be increased by tissue culture and micropropagation techniques, as well as be mentioned in protective screen houses to exclude pathogen and their vectors. Exclusion is illustrated by heat or chemical treatment of vegetative material such as pulp, corns, chromes, and woody cutting to eliminate fungi or viruses that are established within the plant material. Second strategy in plant disease control is pathogen eradication and reduction of inoculum. This principle aims at eliminating a pathogen after it's introduced into an area, but before it has become well established or widely spread. It can be applied to individual plants, seed lots, field or region, but generally is not effective over large geographic areas. Eradication involves removing infested soil, fumigating soil in infested field, removal and burning of diseased trees, sanitation, crop rotation, eliminate weeds and alternative hosts. Use techniques that disfavor factors and movement of the pathogen, and soil sterilization. And the end also we can use the biocontrol as one of the means to reduce the pathogen in this infected area. The third strategy in plant disease control is plant protection. These principles depend on establishing a barrier between the pathogen and the host plant or the susceptible part of the host plant. It is usually thought of as a chemical barrier, for example, fungicides, bactericides, nematicides, but it can also be a physical or temporal barrier. Protection often involves some culture practices that modifies the environment, such as tillage, irrigation, or altering soil pH. It may also involve changing date or depth of seeding, plant spacing, pruning, and thinning, or other practices that allow plants to escape infection or reduce severity of disease. One of the important strategies to control of plant disease is chemical management. For example, fungicides. Fungicides have been used for more than 100 years, and new fungicides continue to be developed. Some fungicides have narrow range of activity and are used primarily for control of specific groups of disease, such as downy mildew, rust, smuts, or powdery mildew, while others are active against a wider range of disease. Fungicides can be applied by any of several methods: ground sprays, airplanes, or through irrigation system, but to be effective application must be done properly. First, the fungicide must be legally registered for use on the plant involved and against the target disease. Several different chemicals may be registered for the same group or disease. If the different fungicides are similar in effectiveness, cost, and ease of application and safety, then timing of application becomes the most critical factor. If applied too early, much of the chemical will be wasted before it can be effective. If applied too late, it will be largely ineffective. Distribution of the spray droplets is important. The finer the spray, the more complete the coverage on the plant surface. However, very small droplets form a mist that is easily displaced by wind. We have different types of fungicides. Some of them play as protective, that means application prevents the establishment of an infection, or curative, and application interrupts the development of an established infection before visible symptoms. Some of them used as eradicant, and that means application interrupts further development of an established infection having visible symptoms. Residual remain on surface of the leaf and provide protection. The last type is systemic, and that means it movement of fungicides inside the plant, some of them locally or throughout the plant, that means systemic of fungicide. We have two main classes of fungicides: inorganics and organics. Inorganics fungicides are protective fungicides and including some simple elements like sulfur or metallic compounds of copper or mercury. The famous one known as Bordeaux mixture, a basic copper sulfate fungicide, was the first widely used fungicide and is still used today in various forms. These inorganic fungicides have the advantages and disadvantages. For example, it's inexpensive, widely available, and minimal threat to environment. But their disadvantages are phytotoxic at temperature above 85 Fahrenheit, and overuse may result in development of pathogen resistance. The second class is organic fungicides. Organic are protective fungicides and has broad spectrum control against different fungal disease, for example, serum and Aq10. Last class of fungicides is fumigants. Fumigants used for soil fumigation. Soil fumigation has been widely used eradication strategy. This technology involves introducing gas-forming chemicals such as carbon disulfide and methyl bromide into soil to kill target pathogen. The last strategy can apply to control of plant disease is biological control. Biological control involves the use of one living organism to control another, and this management technology has received much attention in recent times. However, the number of biological agents registered for use is relatively small, and success has been limited, and the application has been largely restricted to intensively managed high value crops such as greenhouse plants. After we know the definition of biological control, we can answer the question, is biological control biotechnology? Yes, of course, because biotechnology is a set of tools that utilize living things to solve a problem or to produce product. And when we talk about the beneficial microorganism which can use to suppress or inactive the action of the pathogen, that means biological control is one of the tools of biotechnology. We apply the biological control in two ways: by direct antagonism against the pathogen, or indirect effect on the pathogen. Both ways depend on destruction of the propagative units or biomass of the pathogen, prevention of inoculum formation, and weakening or displacement of the pathogen in infested residue. The first way for the biological agent to affect on the pathogen, direct antagonism. The biological agent can use the three mechanisms to affect on the pathogen. Competition, and that means biological agent can more efficiently utilize space and nutrient than pathogen. The second, that means antibiosis. The biological agent can produce one or more compounds can affect directly on the pathogen, for example, antibiotics. The last mechanism is parasitism. The biological agent utilizes the pathogen as a target for food or for reproduction and can make completely death for the pathogen. The second way can the biological agent affect on the pathogen is indirect antagonism during or through induced resistance. Induced resistance, that means the biological agent have or can indirectly stimulate the plant to be resistant. For example, do you hear about the beneficial microorganism or symbiotic association? Mycorrhiza fungi, nitrogen fixing bacteria, plant growth promoting rhizobacteria. All of these beneficial microorganisms can stimulate the resistance in plant, and we know that as induced resistance. That means the biological agent can affect on the pathogen in indirect method, as it acts as a barrier for invading pathogen by providing antagonistic chemicals, by competing with the pathogen, by increasing the nutrient uptake ability of plants, and the last by changing the amount and type of plant root exudates. At the end, we can find now in the market some pesticides known as biopesticides. Bio, this part means involving life or living organism. Pesticides means includes substances or mixture or substances intended for preventing and destroying and controlling any pest. Biopesticides together refers introduction of any living organism such as microorganisms including bacteria, fungi, nematodes, virus, protozoa, and parasitoids that control pests by biological and non-toxic means. For example, trichoderma, bacillus thuringiensis, and very different type of beneficial microorganism. One of the biopesticides in the market is Aq10 Biofungicides. These include biocontrol organism Amplomyces quasquaris isolate M10. This biological agent can affect on the powdery mildew during or through hyperparasitism and direct penetration. We can apply these biofungicides on different crops, apples, curcupids, grapes, ornamental, tomato, strawberries, and we can use as water dispersal granules and application as spraying on the infected plant. The advantages of biological control include this way is more specific for different species of the pathogen and safe for environmental and human, and we can avoid using a chemical resistance and residue for the pathogen. But these advantages, hard to get as soon as possible, must be used immediately and cannot store, may need more than one species. Watch environmental condition because it's actually living organism and can affect by the environmental condition. Expensive because we have a little bit information about the species can use as a beneficial microorganism and as a biological agent in biological control.